Welcome to our video on protoplast fusion. Today, we'll explore this fascinating technique used in plant breeding and genetic engineering. Good day viewers, a warm welcome once again to Biochemistry Tutor 1. If you are new to the channel, like and subscribe. Hit the bell icon to get personalized notification as soon as we drop amazing content like this. Let's dive straight into today's topic. Imagine a world where plants can be designed to thrive in any environment, where crops can be engineered to feed the world, and where the boundaries of nature are pushed to create something entirely new. But what if we told you that there's a hidden limitation that's been holding us back? A barrier that's prevented us from unlocking the full potential of plant breeding. We're talking about the crossability barrier, a natural boundary that restricts the combination of genetic traits between different species. But what if we could break through this barrier? What if we could combine the best traits from different plants, regardless of their species or genetic makeup? Welcome to the fascinating world of protoplast fusion, where scientists are doing just that. By fusing plant cells without their cell walls, they're creating new combinations of genetic traits that were previously impossible. Let's dive in and explore the incredible possibilities of protoplast fusion. Let's break somatic hybridization or protoplast fusion down. A protoplast is a plant cell that's been stripped of its cell wall, leaving behind a flexible, membrane-bound cell. Think of it as a cell in its most basic form, free from the rigid structure that normally surrounds it. Fusion, on the other hand, is the process of combining two or more cells to create a new cell with combined genetic material. Now, let's combine these two concepts. Protoplast fusion is the process of fusing two or more protoplasts from different cells or organisms to create a new cell with a unique combination of genetic traits. By removing the cell walls and fusing the protoplasts, scientists can create new cells that combine the best traits from different parents, regardless of their species or genetic makeup. This technique allows for the creation of new plant varieties with desirable characteristics, such as increased disease resistance, improved yield, or enhanced nutritional content. Protoplast fusion is a multi-step process that involves isolating protoplasts from plant cells, fusing them together, and regenerating new cells with combined genetic traits. Here's how it works. First, plant cells are isolated from tissues using the enzyme called pectinase to digest the middle lamellar. The plant cells are plasmolyzed by subjecting them to a hypertonic solution. This will make the plasma membrane shrink from the cell wall, making it possible to cut through the plant cells to release the protoplasts. This mechanical method destroys most of the protoplast, and it's not good enough for the protoplast isolation. As an improvement to this, enzymatic treatment of the plant cells have been proven to yield better results. In the enzymatic method, protoplasts are isolated from plant cells using the enzyme called cellulase that break down the cell walls. This leaves behind a suspension of protoplasts that can be manipulated in the lab. Next, the protoplasts are fused together using chemical like polyethylene glycol in synergism with calcium ions or electrical methods. This can be done using a variety of techniques, such as electrofusion or chemical fusion, to create a new cell with combined genetic material. Once the protoplasts are fused, the resulting cells are cultured in a nutrient-rich medium that promotes cell growth and regeneration. Over time, the cells begin to regenerate their cell walls and form new tissues. As the new cells grow and develop, they can be screened for desirable traits such as disease resistance, improved yield, or enhanced nutritional content. This allows scientists to select and breed new plant varieties with specific characteristics. Finally, the new plant varieties can be grown and propagated using traditional plant breeding techniques. This can lead to the development of new crops with improved traits that can benefit agriculture, food security, and the environment. Why does somatic hybridization really matters? Protoplast fusion is generally essential for somatic hybridization. Allows genetic recombination between sexually incompatible species. Trait transfer. Facilitates the movement of desirable traits like disease resistance, stress tolerance, or cytoplasmic male sterility across species boundaries. Crop improvement. Used in breeding programs for crops like tobacco, tomato, potato, and citrus to enhance yield, resilience, and quality.